Hello students, the next point in your plant tissue culture topic, that is the artificial seeds, or which is also called as the synthetic seeds. So what is artificial seed? These artificial seeds is made up of somatic embryos surrounded by the nutrient medium, which is protected by a thin chemical membrane. So, synthetic seeds, that means it is the somatic embryo. सोमेटिक एम्ब्रियो रहता है. एम्ब्रियो को जो न्यूट्रिएंट मीडियम है, न्यूट्रिएंट मीडियम से कवर किया जाता है, और जो न्यूट्रिएंट मीडियम है उसका जो सोमेटिक एम्ब्रियो उसके साइड में उसका कवरिंग बन जाता है. दैट इज द synthetic seeds. So naturally seed structure, as you know, in that the middle portion is the embryo, then there is the endosperm is present, and on this embryo and endosperm covered with the seed coat. Similar structure is developed in the synthetic seeds, in that at the center there is the somatic embryo, and that is covered by the artificial seed coats. So what is the features of these artificial seeds? The artificial seeds are smaller in size when compared to the same natural seeds. जो हमारे नेचुरल सीड्स रहते हैं उससे बहुत छोटा है हम आर्टिफिशियल सीड्स बनाते हैं. They contains only the somatic embryos. The somatic embryo is protected by an envelope of nutrient medium which is nourishes the developing embryo. जो somatic embryo और जो artificial सीड्स बनाते हैं उसमें जो भी एम्ब्रियो रहता है, सोमेटिक एम्ब्रियो को दिस एम्ब्रियो कवर्ड विथ दी और एनवलप विथ द न्यूट्रिएंट मीडियम. So these nutrients will supply to this, or they will be provided nutrition to this embryo. The seeds have minimum essential amount of food, so the size of the seed is too small. The seeds are bound by a synthetic membrane made up by the sodium alginate or polyacrylamide gel. These synthetic seeds are very small in size, and they are bounded by the synthetic membrane. So the synthetic membrane is prepared with the help of sodium alginate or polyacrylamide gels. The artificial seeds can be stored for years at the room temperature. So we can store these seeds for many years. This can transport from place to place. Then the germination of artificial seed is similar to the natural seeds. So due to the small size, the transportation of the seeds are possible from one place to the another place, and germination procedure is same as like the natural seeds. These artificial seed will be grow. Next is the methods of production. So how actually these synthetic seeds are prepared or produced, you can see. So first, these induced artificial embryos by somatic embryogenesis. The somatic embryo with 2% sodium alginate drop on the embryo in both of calcium salt, that is CaNO3 twice solution for 30 minutes. It results in formations of artificial seed coats around the embryo and sodium alginate. Then these sieve beads through the nylon mesh. CaNO3 twice solution can be recycled. Beads are nothing but the artificial seeds. So a very simpler procedure of the formations of synthetic seeds. These synthetic seeds are nothing but the preparing as a beads. So first of all, we can take the embryos of artificial from the somatic embryogenesis process. If we can got this embryo, then embryo will be uh, dipped in the 2% sodium alginate solution. Then these also drop these embryos on the calcium salt, so they will be for up to the 30 minutes. So the salts or the sodium alginate, the calcium salt will make the coating of these seeds, so we can form the beads like structure of these seeds. And then next point is the types of artificial seeds. The first one is the encapsulated seeds. The seeds individually enclosed in capsule. Each capsule consists of an embryo, some amount of nutrient medium, and sodium alginate membrane. So in encapsulated, that means this capsule like balls will be developed or beads will be developed with the help of sodium alginate. This sodium alginate make the membrane around the embryos. Then second type is the seed gel suspension. So these embryos are placed in the semi-solid gel. As a result, seed gel suspension is formed. If we place this embryo on the semi-solid gel, then these suspension will be formed around the embryos. Then seed type, the embryos are placed in between the two types of semi-solid medium loaded with the sodium alginate or polyacrylamide gel. So in these seed types, we can use the two mediums. First one is the sodium alginate or polyacrylamide gel. So this gel or sodium alginate will be make the capsules or membrane around the seeds. So these are the types of seeds will used in preparation of the synthetic seeds. Then commercial aspects of this artificial seeds. In the synthetic seeds or artificial seeds have many advantages, so it is commercially very important. The first one is they can be stored up to a years without loss of viability. Naturally, if we take the seeds for many years, they will be not all seeds not will be germinate, so they will be not viable. But if we prepare the seeds by the methods of the artificial seeds, these seeds will be remains viable in many years. Then they will be easy to the handles. They can be directly sown in the soil like natural seeds. They does not need hardening in greenhouse. Then from small tissues, many embryos, that is many synthetic seeds can be obtained. Plant raised by synthetic seeds of some callus are identical. It shows no variations to the parental types. It means superiority of the parents maintained in artificial seeds. It is like the vegetative propagations. In artificial seeds, some herbicides, pesticides can be also added in nutrient medium. So there is no need to apply the herbicides during the sowing. So it works very effectively. Also, the extra nutrients may provided in artificial nutrient medium of artificial seeds, so no need to add fertilizers externally. Generally, the plants raised by artificial seeds are disease free. These seeds can transported from one place to other. The nutrient medium of artificial seed, mycorrhiza or blue-green algae may also added to get plantlets. So, a natural seed production, germination, dormancy, etc. problems in some commercially important plants. In such cases, artificial seed production is answers or solutions. So, with these different aspects of the synthetic seeds, we can use commercially these artificial seeds, because these artificial seeds are one thing, they will be remain viable in many years, then it will be easy to handling, then they can be as a natural seeds who can directly sown in the soil. There is the no needs of the hardening, as you know, in various processes these greenhouse sorry due to the tissue culture developed plants, there is a need to, first of all you have to hardening this plant and then we can expose to the market. But these synthetic seeds plants, there is the no need to hardening. Then these from the very small embryoids we can make the number of plantlets. Also, these plants have an identical to its mother plants, and also they will be superior than these mother plants, so it is very useful. In also the artificial seeds are also, there is the adding various herbicides, pesticides, so they can work very effectively, and there is the no need to the plants to supply these to supply the herbicides or pesticides on plantlets. Also, in these synthetic seeds, we can also supply the some nutrients. That's why these there is no need for artificially supplying of the fertilizers. Generally, these plants will having disease free due to they are very healthy or superior plants. They will be almost disease free, and also we can adding some, if we adding some mycorrhizal or blue green algae, they will be beneficial to the peoples. So these on the natural seed productions or seed dormancy, it will be very useful tool. We can use these artificial seeds. So this is all regarding the artificial seeds. So now I stop here.